I'm Catherine Hales from the Departments of English and Design Media Arts, and I'd like to welcome you all here today. This event on genetics and culture is being sponsored by Synapse, which is an interdisciplinary initiative here at UCLA. The acronym stands for Social Interfaces and Networks in Advanced Programmable Simulations and Environments. And the purpose of Synapse, as the name suggests, is to promote interdisciplinary conversation, exchange, debate, and collaboration. And we're grateful to Vice Chancellor for Research Roberto Pache to giving us funding for this year for Synapse, and also to Dean Newman of the School of Arts and Architecture for baptizing us as a small C center. So I'll turn the podium over now to Victoria Vesna, Chair of Design Media Arts, who will introduce our panelists and respondents. And I'll also just mention that this event is being streamed live out onto the web, so we have our physically present participants, but a number of virtually present participants as well.
Thank you. Thank you, Catherine. This is Friday, Los Angeles and UCLA. Everybody disappears or is watching the stream. But we're going to go ahead with our program, and I think I'd like to first start with just pointing to some of the artworks that you see, and some of the students who created this work are actually part of a class that is being taught by Ruth West, who is a graduate student in our program and who has been a geneticist for 8 years before she joined our program. She is trying to combine art and science in a truly interesting way, where metaphor and fact and fiction come together in ways that become really compelling in her own work. But she also teaches a class that is 2/3 biology and students from different sciences, actually, and then 1/3 students from our department. So when you get a chance, take a look a little more at the kind of work that's here from students who had no prior experience actually with art making, but who are engaging creative and cultural ideas from a different kind of active way. These are some images from the class and some of the ideas that they've been working on. This is the class, and Ruth West is navigating this website. She is the person who came up with this. It's a class that's been really full of kind of enthusiastic response, and we're hoping to have more of these kind of classes initiated and on campus that really cross over through practice and not just talking, even though we're talking here today. With that, I'll move on and just introduce one speaker at a time, starting with Gregory Stock, who's the Director of Program on Medicine, Technology and Society, UCLA. And he was kind enough to come and join us. Gregory is someone who's really out there speaking on genetics on many levels, from Congress through all kinds of commissions, and his program has the goal to bring about a public debate on technologies and their implications. He's been a prolific author, and he has a new book coming out called Redesigning Humans, which he's going to talk about today, I believe. I hope. More broadly, same topic. More broadly. In any case, what Greg will do is introduce the subject in a very kind of broad sweep. And I guess the book does the same thing. to a certain degree. So welcome, Gregory Stock.
Thank you. So genetics and culture and art are they form a nice grouping. Basically, because we're trying to come to grips with a lot of the very challenging issues that are arising because of the revolution in genomics and more broadly really in molecular biology that is going on today. And this is going to cause enormous changes, not only in medicine and healthcare, but cultural changes, and these are challenging us in spiritual and philosophical ways too. And to see sort of why this is occurring and how it's occurring, I think we need to look at the big picture, which is what I want to give you a feel for as a way of serving as a foundation for other more specific discussions that we'll be having. So it's been about 2 years now since Francis Collins and I guess it was Collins and Venter announced the rough sequence of the human genome. And, you know, not a whole lot has happened. Everything's, life is pretty much going forward unchanged. And so you might think, well, this is not really such a big deal, or is at least not as the extravagant metaphor that you may have heard at the time. Things like, here we're unraveling the code of codes, we're reading the book of life, all of these sorts of things. Well, maybe it isn't such a big deal after all. But I want to show you that really it's just kind of the calm before the storm. And the reason for this is that genetics is at our core. And as we learn not only to unravel the human genome, but we learn to manipulate our genetics, we're really learning to manipulate ourselves. And technology can have enormous impacts on our lives. You just have to go out and take a walk in any major city in the United States to see that. The United States doesn't have a very Los Angeles doesn't have a very developed downtown, but still, you walk downtown and you're in these valleys of concrete and steel and glass. This is nothing like the stomping ground of our Pleistocene ancestors. And what I want to suggest is that as we begin to manipulate our biology, I mean, what really has happened is that this technology that we've applied to alter the world around us has achieved the precision and the power to be able to be turned back upon our own selves. And I want to suggest that we are probably going to modify our own selves every bit as profoundly as we've changed the world around us, and that these sorts of changes are probably going to become very rapidly, certainly far more rapidly than the changes to the external world around us, and that it's going to revolutionize medicine and healthcare. It's going to just remake broad swaths of the economy. I won't be talking very much about that. It's going to alter the way we manage our emotions potentially. It will change the way we have children. It will potentially change the human lifespan. This is really big stuff. It's going to bring into question the very nature of what it is to be a human being. And I think that that's why it's, you know, obviously going to generate huge cultural shifts, and why it's obviously a focus for art, at least much more so than just the visual arts. I mean, art is trying to was going to both reflect these changes, and it's going to be a way of grappling with the way these changes impact our lives, our images of ourselves, our society, and sort of to render these changes into human terms. So I guess the real question is, what subject would be better than art? I mean, what subject would be better than genomics for art and for dealing with these issues? So I want to take an example, a recent one that you've all heard about, cloning. I mean, here's something that since the creation of Dolly, we have Gene and the first cloned cow. Now we have CC, the little cloned calico kitten. You know, and every time there's this huge story and everybody gets very concerned, and there's a lot of hand-wringing of when is this going to happen in human beings, what do we need to do about this? And to me, this is an example about the way these technologies can scare people and can have impacts that are really more a question of sort of projection and philosophy and theology than they are about the realities of what's likely to happen anytime soon. I mean, it's gone so far that in next month there are going to be votes in the U.S. Senate on bills not only to ban reproductive cloning, but to actually criminalize basic research directed towards Parkinson's and diabetes and Alzheimer's and other diseases, a whole realm called regenerative medicine, that the technology of which might overlap with reproductive cloning. So there is a bill that was passed by the House of Representatives last year that would make it punishable by 10 years in prison for a scientist to transfer the nucleus of one cell into a human embryo, and punishable by $1 million fine as well, and that it would be equally illegal to import the product of such manipulation. So if someone went to Britain, say, where such regenerative medicine might be legal and got treatment and then came back to the United States, they could be thrown in prison for 10 years. So this is really sort of harkens back to Galileo and his trial and these sorts of things. And so these cultural sort of philosophical fears are really obvious, but you've got to put it in context of what's really going on. There have been thousands of attempts to do nuclear transfer in rhesus monkeys, none of which have been successful. It's undoubtedly going to be too dangerous to do in humans for a while. It's quite likely that it will happen in 5 to 10 years. And so what? I mean, this is the creation of a delayed identical twin, which is hardly, it's a little strange, but it's not going to shake the foundations of our understanding of ourselves, I don't think. Same thing with designer children, where there are enormous projections that essentially evoke eugenics and Nazi concentration camps. And so we imbue these technologies with incredible cultural amplifications. And I think that we'll talk about those more specifically in a little while, probably. Before we do, though, I want to just mention the big picture, the Human Genome Project, where we are. We have essentially identified the genes, a list of genes, although there's some questions about the details of that. And we all have the same genes. We're all Homo sapiens. We have a library of the variants, the flavors of each of those genes, the common flavors, the so-called SNPs, or single nucleotide polymorphisms. And those are what the variation among us in which of those flavors we have is sort of what constitutes our biological individuality at a core level. We have things called DNA chips, which are allowing us to read those genes, or at least identify which of the common variants we have, and do so much more cheaply, or increasingly cheaply, and bringing up the issue of what it'll be like when we have essentially a storefront genome, where you can go out and get your full genome for somewhere between $100 and $1,000. And in fact, that's going to be the topic of a symposium here on campus in the fall called the Storefront Genome, which tries to look at what the implications of that are. So you have the genes, you have the common flavors of the genes, you have ways of reading out those, and we have the computer technology, bioinformatics as it's called, to be able to make sense of all this deluge of information. And it's going to have enormous implications when all this comes together, in that we will be able to interpret a lot of what our genetics means about who we are. And genes are very important. They don't tell the whole story, they're not our destiny, but they do tell us our risks, our predispositions, and a good rule of thumb, which comes not from genomics but from twin studies, identical twins that are raised together or raised apart, is that most of the things that you think are important about who you are, whether you're shy, how tall you are, what talents you have, what disease risks you have, the variation in the human population for those individual traits, probably about 1/4 to 3/4 of it is determined by genetics. And the rest of it is shaped by your own unique personal experiences in life. and whether you get screamed at, what your fetal environment is, all these sorts of things. Very little, it turns out, is shaped by shared family environments, the kinds of things that we direct social programs towards. So the implications as we begin to get all this information are that it's going to have a lot of near-term implications on medicine. It's going to reshape healthcare, as I said. There's going to be a huge swing towards preventive medicine as we begin to find out what our risks and vulnerabilities are. Well, we're going to want to do something about it. We're going to want to intervene pharmaceutically. We're going to want to intervene with lifestyle changes that we might not otherwise make. That's going to be a big change. There's going to be an issue of who has access to this. And it's not just who could get access, but who can understand it. How do you communicate all this information to people when physicians themselves are not going to be able to understand the details of it, don't have the time for that, and when it's extremely complicated information? Will it be delivered over the internet? There are only 2,500 genetic counselors as it is in the United States today, so that's a big issue. Another is who's going to pay for all these preventive measures, for all these elective procedures, and how much do we in fact want to pay? Because there are trade-offs, and yet we're going to be spending more and more of our GDP on health care, but how much do we really want to do? Because health is very important to us, clearly, but it's not everything. And then political issues, because we're going to have to come to grips with our differences. There are, much as we'd like to say we're all the same, you just have to look around and you know that we're different. And a lot of those differences have genetic underpinnings. And so we're going to have to come to grips with these differences, and they're differences not just between us as individuals, but differences among groups, and do that in the context of an egalitarian society. So those are the short-term kinds of things, and they're going to be quite adequate grist for art and cultural changes. But if you get to the longer-term things, it's tremendous. We're going to begin, I believe, to have the ability to manipulate our emotions in various ways that are quite precise. Certainly, Ritalin and Prozac, those sorts of things, are just the clumsy first steps. What would it be like if you had a drug that you could take that made you feel really contented and fulfilled, just happy to be alive, and didn't have any real physiological side effects? Could you resist taking that? And if you didn't, then what would motivate you to do what you do, to make choices about how you would live? And who would we be? So that's a whole big area. Another area is reshaping ourselves, actually manipulating our biology in very profound ways. And I don't mean growing new gills or an extra pair of arms or wings or anything, but take something like human lifespan. If we unravel the genetics of aging, and mainstream researchers in the biology of aging think this is quite plausible, that basically the first person who will live to be 150 years old is already alive. And so that's a big area. Another is human reproduction, designer children, if we can understand what genetic means about who we are, and we can read the genetics of an embryo, which we can already begin to do, then we're going to want to make choices about who our children are. It seems rather obvious to me that this is the case. And this will move from disease screens to looking at lesser vulnerabilities like risks for being manic depressive to simple choices about personality and temperament. And this will become very powerful, and in fact we can talk about that in questions and answers, but I don't want to get into the details right now. So anybody who wonders about the relevance of genetics for culture and art just has to step back a little bit. It touches upon our emotional nature, upon human reproduction, which is something very near and dear to us all, upon aging, upon our health and vitality and our sickness, upon access to these technologies and the balance of resources within society, upon our relationship with others, including our children, including whole other generations, upon the randomness of life, and upon theology and philosophy. And it seems to me that sort of covers the whole ball of wax pretty much. And these issues are going to come up very rapidly. So it's really not a question of if these things are going to happen, if these technologies will be used, it's when and how they will be. And humanity is definitely going to go down this path, and it's going to do so for two reasons. The first is because you actually don't need to even pursue them for them to happen, because they're a mere spin-off of mainstream research in biomedicine in areas that we all really care about a lot. And the second reason, which is, I think, more interesting, is it's because we're human. We tinker, we explore, we try to modify ourselves, we try to improve our lives in any way we can. And ultimately, the issue of enhancement technologies will revolve around what I think is the truism is that essentially lots and lots of people would like to live longer, they'd like to be healthier, they'd like to be more talented in one way or another, and if they can't have it for themselves, they'd certainly like to give these advantages to their children. No matter how much we debate about these things, there is not going to be a consensus about them. The next frontier is not going to be space, it's going to be ourselves, and it's going to be quite an adventure that we're embarking upon. These things touch our values too deeply for us to agree upon them. The coming struggles, I believe, are going to make the abortion debate look relatively tame, and it's going to be a battle between people who see all this stuff that I'm talking about as the invasion of the inhuman, absolutely the worst thing that could happen to us, something that is just iniquity, and that is something we should struggle against with everything we can, in every way we can, and other people who say, this is the flowering of humanity, this is wonderful. This is our chance to do things to overcome limitations of our own biology, perhaps even elements of aging and things of that sort, that other generations have only dreamed about, and they're going to reach for it in any way they can. So genomics and all of the implications of these fields are going to really challenge our vision of who we are. They're going to try our willingness to see an open-ended future. And the challenge is not going to be how we deal with cloning or stem cell technology or genetically modified foods or any of these individual technologies. We'll probably muddle through just fine. It's going to be whether we have the courage to face an open-ended, uncertain, unclear future that is going to be profoundly different from the present, or whether we're going to pull back out of fear, probably, and try and stop these developments and turn their development over to other braver peoples in other regions of the world. And here we are at the beginning of a new millennium, and I think at the end of this millennium, when future humans look back at this era, they're going to see it as this challenging, turbulent, difficult, very confusing moment in time. And they're also going to just be in awe. They're going to say, this was this extraordinary instant in time, this glorious moment when we laid down the future of the foundation of their lives. And they're going to look at it as this extraordinary time. And I think that a cornerstone of that future is going to be the reworking of human biology. And it's going to come rather gradually at first. It's going to pick up speed as the sort of fruits of the genomic technologies and all of these other associated realms begin to bear fruit, as they begin to bear fruit, and ultimately it's going to bring us to the question of what it is to be a human being. And I think it's a remarkable privilege to be able to be here just to observe this occurrence, which is extraordinary, not just in the history of human life, but in the history of life as a whole. And what's challenging about it is that we're not just observers, we're the architects of this, we're making it happen. And we are the objects of it because it's us, we're the ones that it's acting upon, and also we are the interpreters of all this. And that's really what we're going to be talking about here today, because we're the ones that decide the value in this and what it all means. And I just want to close with a quote from 430 BC from Thucydides, who I think quite eloquently captured our call today. He said, "The bravest are surely those who have the clearest vision of what is before them, glory and danger alike, and yet notwithstanding, they go out and meet it." So thank you.
Our Our next speaker is Stephen Wilson, who's walking over to the podium. I should just mention that we're just going to power through, everybody giving a 20-minute presentation, take a break, and then come together. Since we're a pretty small group, we could break this up and make it into a more closer discussion. So after the talks, we'll do the questions and answers. Stephen Wilson is San Francisco-based author, artist, and professor who explores the cultural implications of new technologies. He is the head of the Conceptual Information Arts program at San Francisco State U and has been an artist in residence at Xerox PARC and Entity Research Labs. He just released his new book called Information Arts, published by MIT Press, where he discusses the research of artists and the new evolving field that involves inquiries in fields such as biology, medicine, physics, artificial life, telepresence, body sensors, artificial intelligence, and of course information systems. We invited him today to give us a broad survey, once again, to follow Gregory's broad issues and some of the rhetoric that we hear about genetic research, some of the issues that are raised by the research and what it means to humanity, to Stephen, who will give us a broad spectrum of how artists are responding to some of these issues. So please welcome Stephen.
Thanks. Is this on? I guess it is. I think some artists would be very comfortable with what Gregory said, and some would be very uncomfortable. So I think we could characterize the arts as kind of passionate and ambivalence. So what I want to do today is I want to talk about what artists have been doing in the realm of genetics and bioengineering, and then extract some issues to think about, and hopefully that'll be useful for the discussion. And the first thing is that artists approach it in a great variety of ways. Some want to critically comment on science, and science, the whole context of science. Some want to comment on corporate and governmental control of bioengineering research. Some are concerned about eugenics. Some are very interested in issues of identity in the body. Some are very curious about the new possibilities, and they want to work with this new stuff. Some want to modify their own bodies. And some are just drawn in by the lure of creation. And I think genetics and bioengineering is a tremendously important realm for new creation. So I want to just quickly go over some historical trends that relate to this. First of all, the arts have imagined alternative life for a long time. So there's a long history of mythical creatures, dragons and Pegasus and unicorns and whatever. And I think that's part of what's going on as artists move into genetics and genetic engineering. There's Bosch. I don't know if we wanted to meet some of his creatures, but certainly worked with it. More recently, there's a French artist, Louis Beck, who 20 years ago developed the idea of hypozoology. And his notion was that one of the things artists could do is imagine life forms that don't exist yet. And when computers came, then he would use computers to visualize them. So I think that's part of the background here as artists begin to move into genetic engineering. Now there's some issues of working with the living, with living materials. Here's some examples. There's all kinds of folk arts around the world of working with animals and plants. In the 60s, artists, performance artists, installation artists began to incorporate live animals in their work. This is a picture from Joseph Boy's piece, I Like America, America Likes Me. Spent a week with a coyote. There's a whole tradition of ecological art. Newton Harrison, Helen Mayer Harrison, trying to work with large environments involving scientists, political, governmental, and the public. But there's some issues in this. Why haven't artists worked more with living materials? And one is issue of artistic control. Living materials have a mind of their own. And so that's been somewhat problematic for a lot of artists. There's issues of ethics. I mean, are the animals, I always wondered about Joseph Boy's coyote, what the coyote thought about that performance. There's issues of commodification. You can't commodify this stuff very easily sometimes. So that stopped a lot of artists from getting involved. And George Jessert, who I'll talk about before, noted that museum architecture is designed to keep life out. There's no light, there's no water, there's lots of reasons that it's... so when he did his plant-based stuff, it was very hard to do it within the museum context. I just want to note quickly, there are other kinds of biological work that we're not going to talk about today. Manipulations of the bodies, biosensors, medicine, ecological art. I mean, these are all kind of interesting areas where artists are working with biological materials. But we're not going to talk about those today. We're going to talk primarily about genetics and genetic engineering. All right, so some artists have worked in historic forms, breeding. I mean, there's a long history of us doing genetic engineering and farmers breeding certain kinds of animals and crops. Matter of fact, I heard a news item a few months ago that the genetic engineering folks were trying to get the same rules applied to breeders that are applied to them, because saying essentially breeders are doing genetic engineering, so that there should be the same kind of restrictions on what they do. So this is George Jessert. He's been working for 20 years with irises. He breeds amazing varieties of Pacific Coast irises. Some of them monstrous, some beautiful, and he does it within an art context. He's done some piece where he has the audience. He did some pieces with Coilus where the audience could make some decisions, and that would make the decision for the next generation. More recently, Christoph Ebener and Yuli Winters did a piece at Arts Electronica called Bite. They bred mice to eat computer cables. And then they would display, you can see here's the mice slide up, and as they ate the cables, the computer displays would change, and you could experience this breeding outcomes. Adam Zaretsky is a young artist, worked with Eduardo Koch, spent a year at MIT. He's working biological materials. The most recent thing he did is a Humperdinck effect. He worked with genetically modified bacteria, E. coli, and he played different kinds of music to them to see how it would affect their production of antibiotics. And it turned out that I can't even say Humperdinck music was the best for producing antibiotics. But I mean, just think of this as a kind of, so the researchers at MIT were quite shocked, he said. Andrea Zettel did a piece a while ago where she had chickens, and in order to get to the place where they could lay the eggs, they had to get through these barriers. So she was looking at which the setting tried to breed the best flyers among the chickens. Now, more recently, there are artists actually working with contemporary genetic engineering. Probably the most famous at this part is Eduardo Koch. Artist teaches at the Art Institute of Chicago. He did the GFP bunny, which caused a great stir in France. He commissioned some for genetic engineers to put in the jellyfish fluorescence gene into a bunny. And that bunny was born. His plan was that bunny should come home with him, but the government impounded the bunny, wouldn't let him have it because they decided that wasn't an appropriate kind of use of genetic engineering. I mean, it's more complex than that. But so he has a worldwide movement to see if he can get his bunny back. And one of the interesting things is when he does his work, he wants to live with the creatures that result. And he feels that's an important kind of statement, that it's not just something that's done to the bunnies, but something that the artist has long-term commitment to. His most recent piece is this 8-day. It's a biorobot. There are several kinds of fluorescent animals in this environment near the robot. There's a fluorescent amoeba here, and then people on the web can come in also. So what the robot does is jointly determined by the reactions of the amoeba and people on the web. Critical Art Ensemble, I'll talk more about them and kind of critical approaches toward all this, has done a variety of pieces. The one I want to talk about here is they brewed some transgenic beer. They transferred part of the human genome into the yeast of the beer. And you know, Paul Van Naus is one of the artists, and he took out one of these beers and offered it to me, and it was a very interesting moment, because you know, it wasn't something you were going to just swill down, really. I didn't drink it, ultimately. But we'll talk more about why didn't I drink it. Oops. Moving on to some others, there are some artists who have embedded messages, uses genetic engineering to embed messages into creatures. Joe Davis is an artist, researcher at MIT. He's been doing this for a long time. He did the microvenus, where he embedded this rune, which he felt was a basic kind of human image. Came up with a method to embed it in the base sequence in the genetic code in the E. coli. So he likes to say that there are more copies of his artwork than any other artist in the world. There's billions of copies of it. And Eduardo Koch did another piece similar. He took this phrase from the Bible, from Genesis, again, worked out a scheme to change it into Morse code, then embed it into the bacteria. Okay, I'm racing through these. This is on the website. I have a website. I think it's on the Synapse website, the link, so you can read about these. You can follow them up in more detail. Natalie Jeremchenko did, I think, a very wonderful project called One Tree. I think she was in LA recently, so maybe some of you heard her talk about this. She had 1,000 exact clones of the same tree. And then she's going to plant them in different environments in San Francisco. So these things that have genetically identical backgrounds are going to be subjected to different places in San Francisco. And she has she's going to try and monitor them all on the web and monitor their environments and just really explore this notion of genetic background and environmental influences. There's also going to be artificial life trees growing on people's computers. Some artists who are interested in using genetically modified materials to create images or installations. For instance, David Kremers, who's based at Caltech around here, he has worked with genetically engineered, he paints with genetically engineered bacteria that then produce particular colors, and then he fixes the colors after they produce it. So it's and you can see this is a very different approach than someone who's trying to embed some messages in genetic code, and just as you'll see, the range of what artists are doing with this. There's another group in England, a couple of artists in England, Heather Ackroyd and Dan Harvey. They got interested in sun painting. You know, when you put something on the grass, the sun shines on it, that place that's under the grass takes on a different color. Well, it turns out there's now some genetically modified grass that holds its color. The chlorophyll, the stuff that's green, holds its green. So they've been exploring painting pictures with this genetically modified grass. A lot of artists are interested in the schemes of representation. How do we represent the genome? How do we represent chromosomes? Suzanne Anker is a New York artist, been working on a while, working for several years on this. She does sculptural installations where she exposes the chromosome kind of representations and builds installations out of them. And then just to remind us that there are all kinds of decisions that are made about how scientists represent genetic code. And this is just a quick grab of some of those mapping systems. And I think you'll see a lot of activity in the future of artists kind of reflecting on genetic mapping conventions. A lot of artists, and wondering about where does identity come from? What part's inherited? What part comes from environment? And what does this whole thing of the genetic engineering give us access to some of these things, just like Greg was talking about? So some artists are getting ready for that. Mihail and Kim Trong have the genetic gene harvest machine. Not a machine. They've gathered hair samples of creative people around the world and they're storing these for when the DNA becomes important. Nino Manglano Oval has again took the DNA readings of various people and families and arranged them in a display so you can see the family relationships of where these are, of what they look like. Just how to read this new kind of iconography. Paul Van Aus, his artist, he's at Buffalo right now. He's Jamaican, and he did this piece called the Relative Velocity Inscription Device. And what he did is he took the DNA from various people in his family with different cultural backgrounds, and he uses electrophoresis. Now scientists use this when you take genetic material, you subject it to electric charge, some of it moves different amounts based on certain qualities of the DNA. So he's doing he's built the world's largest electrophoresis device, and he's seeing how far the different parts of his family get in this thing. So it's kind of playing on the horse race and just genetics and cultural genetics. Kevin Clark has been doing a series of portraits for a while where he gets the DNA from various famous people and then creates portraits, and he embeds, you can see down here, he embeds the DNA information in the portrait. Gary Schneider did a piece called The First Biological Self-Portrait, where he took every kind of biological material about himself he could, DNA, blood samples, and he created a series of photographs that represent who he is. Here's some music. John Donne and Marianne Clark. John Donne's a musician and Marianne Clark's a biologist. And they took a scheme, and there's several artists working on this, taking the DNA sequence, converting it into sound or music, and in some non-simple way. And Clark has an interesting paper that she finds it very useful as a research device to listen to the different DNA sequences. It's not actually DNA, it's DNA then to the protein sequences that the DNA generates. So here's a little music based on alcohol. Are we? It gets louder here. Maybe that's where they got it. So if you're interested, they have a website full of different DNA of different materials. Then we move into, and I think in some ways this is the most active area right now. Artists around the world are very uncomfortable with the speed with which things are moving and who's controlling where engineering gets done, and trying to find powerful visual performance methods of making that, exposing those issues. So the Critical Art Ensemble has done a series of things. They've done The Flesh Machine, The Cult of New Eve. In The Flesh Machine, visitors come in, you get blood taken, they read your DNA, you're asked questions whether you can be a donor, and then if you pass, then your blood gets frozen for the future. And it's done very much in a fake corporation kind of situation. Natalie's going to talk more about her work, I assume. Coming down, the Creative Time is a group in New York, done a series of pieces called DNA Aid, some billboards, some coffee cup holders, a variety of ways just to bring this information out, get people thinking about it, find visual ways to present it. Alexis Rockman is a painter, paints these creatures of the future and mute creatures. Nancy Burson did it's called a genetic machine, I forget the name of it, but the race machine. and it generates different faces of different races based on the same material. And Natalie's going to talk about, you're going to talk about Metapet, I hope? A little. Okay. Hex Terminators is a group in San Francisco. Actually has some of my ex-students in it. And they go out in the street, do street theater. What I like about what they do is they read as much as they can about the material so that their interventions are based on knowledge of some of what's going on. They did a great piece. They study, in the United States you're not allowed to indicate which foods are genetically modified. In Europe you have to indicate it. So they studied that, and they went to grocery stores, planted giant signs in the food that was genetically modified, just to get people thinking about it. And like I say, there are lots of other projects that people should be aware of, and those are listed. Some photo essays of research settings. Catherine Wagner has been doing photo essays of science and science environments. And within the tradition of photography and photo essay, but some of it's very moving actually to see it. And this one is she has a freezer with samples of various kinds of diseases. Catherine Chalmers did this piece on transgenic mice. I've read about transgenic mice, but to actually see these guys like this really is that's the place where the artist can do something that the essayist has a little trouble doing, I think. And Michael Spano did the Scientist at Work piece. I think that artificial life is a related area. I'm not going to talk about that much, but it is related. It's people looking at silicon-based life, and gets into a lot of things about breeding and competition and evolution. So I encourage you to look at that. But I want to get to extract some of the things I think will be useful for our discussion today. So here we go with passionate ambivalence. And as I look at this stuff, it makes me ask certain questions. And one of the big questions I think artists have to ask as part of the larger cultural community is bioengineering something that the community ought to pursue? And there's an article by Bill Joy recently saying that we're now moving into technologies. Bill Joy is a scientist at Sun. He wrote an article, a lot of people talking about that there are 3 technologies he says are dangerous in a unique way: nanotechnology, genetic engineering, and artificial intelligence. And what's different about all these is that you create something that then runs loose on its own and generates its own progress. And he was saying we really ought to think a little bit about, do we want to go down that line? And I think a lot of artists are asking that question. Now, if you do want to go down that line, there's some interesting things that come up. Adam Zaretsky was a visiting artist with us last semester at San Francisco State. And some of these became very obvious. He had the students submit proposals to every university has a committee on the protection of animal subjects. So he had his students write up research they want to do, art research, and submit these to the research committee. And it's very interesting. First of all, they didn't want to look at them at all. They said, "You're artists. You're not doing anything that you can't be doing animal research." That was their first response. They wanted to refuse to look at it. And then that made it clear to me that, if you're going to do this and you feel there is some risk, what are the hierarchies of value that decide whether something's worth it? If you look at the guidelines for protection of animal subjects, first question, what is the scientific value of what you're doing? Another question is, how many people is it going to benefit? Your prediction of that. So it's interesting, there's a hierarchy: science, medical, commercial, and art's way down at the bottom. And it seemed very hard for this committee to wrap their minds around the idea that art might be a reason for people to be working with animals. The students in that class weren't doing any genetic engineering, but I could see the same thing come up if they were proposing that. Then that made me think about, who owns biology? Why is it that the scientist necessarily, or science as a community, is the one who should be deciding what's valid research for a culture to do? It seems, and this goes with what I think Greg said, it's a much larger question. But we seem to accept that it looks like research, it looks like scientific research, that scientists ought to be making that decision. But I just want to ask that question. They don't own biology. We all own biology. But I don't know where that leads. Because then we get to the next question. Scientists are pretty uptight in a good way about kind of guarding what they do and submitting it to consensual community kind of oversight. Whenever you're going to do something, you have to submit it to, well, there's some rogue scientists too. There aren't artistic traditions like that. Artists don't submit it to other artists and say, is this a good thing for us to be doing? So that's an interesting question. And then I started, here I am, I love art, I've been doing art, my students do art. Would I trust a bioengineering artist as much as I would trust a young scientist? And arts have this tradition of impulse and passion and going down whatever crazy lines of interest they have. It raises some questions when you're doing it in bioengineering. Another danger, I think there's a danger that artists set themselves up only in the oppositional critical position. As I said, the most activity in the art community right now is this critical stance. This is bad stuff, or let's look at it, I think it's bad the way it's dominated by corporate and governmental kinds of forces. But I think art's got to be a little more open than that, and that there are other options. And I talk about this in the book a little bit. I think art could be an independent zone of research. There are lots of things to be doing with bioengineering besides those things that are going to maximize someone's profit. And even lots more to be doing even than are going to develop new medicines. So that's that's a place that I argue that if you had skilled, knowledgeable artists, you would get kinds of research that you might not get out of science or commerce. And just to give an example of Monsanto's trying to sell their Roundup around the world and dominate the crop production. So there's a group of several Asian countries that put together their own biotechnology network. And they want to create better rice, more nutrient, and they want to give it away. So that's, it doesn't only have to be done for commerce. So this is just an example of some research being done in a setting other than maximizing profit. Some young artist contacted me. She wants to make cactus, and the little hairs, she wants to be human hairs. So she wants to see if she can do some genetic engineering so this cactus generates human hairs. It's a delightful idea, but then you start saying, geez, should a lot of effort go into that, and what's the value of that? So we're at an exciting moment. I don't have answers to those, but those are the questions we're going to have to start asking. How many people would like to see a cactus grow human hair? All right, we got a few here. The danger... What? I'm sorry, I can't...
That's the new generation speaking.
I think there's a danger of restriction of media. The most comfortable place for the arts to work is in generating images, generating performances, looking at the visual iconography. But there's this cross currents in art. There is a whole realm that's abstract, representational, imaginary, virtual, critical. And there's another realm that's sensual, embodied, gooey, physical. And so what are you going to do if you're a bioengineering artist and you want to work in that gooey, physical realm? So all I want to say there is there's a danger to just restrict yourself to that media where it's easy. And so as I argue in my book, I'd like to encourage artists to learn the skills to do these things. That if you're interested in bioengineering, then maybe you should be actually doing some things, trying to do something. And I think Natalie's project is tremendously strong in that sense. When I first went there, here's 200 trees. She had to get cloning done, actual physical cloning, and they displayed it at Yerba Buena Center in San Francisco. Here were 200 of these trees, exactly same. They were little seedlings at that point. Exact genetic inheritance. They were already different. That's very powerful. See 200 of these guys put in the same kind of earth with the same light as well as, could be standardized. And they're already beginning to look different. Now, you can write essays about how, genetic inheritance is only part of it and give statistics, but that's the place where a visual artist, I think, can do something very unique. Danger of mystification. There's this, I would urge artists to be wary about that this is too complex. Don't worry your pretty little mind about this. Access is a barrier to investigation. You can't get hold of the stuff. And I think we have a tradition. When I started computers 20 years ago, the exact same rhetoric. The computers were only available in big research universities and government institutions. The notion was if you were an artist, you couldn't understand the coding, you couldn't do the programming. And the notion, it was too expensive. It used to be to run it. It was hundreds of dollars for a second of time on the mainframe computers. And so the same kinds of things are being said now about why artists shouldn't get involved with bioengineering. I said, don't accept that. If it's important to an artist, there are ways to develop access. As a matter of fact, I hear you're going to open a bioengineering wing of the Media and Arts Department, right?
We're moving into the Nano Institute.
And finally, all right, so what do you need to learn? And that's a conundrum also. How much, I don't want artists messing with a gene splicing machine if they don't know a little bit about what they're doing. So yeah, they ought to, if you're going to work in that realm, you have to educate yourself. But then the question is, what constitutes that education? How much? How much do you need to know? And then it's when you get a science education, you don't just learn the skills, you also get socialized. I think you get socialized to realize that to grow hair out of a cactus is a bad idea. And that would be a shame. So the question is, as artists move into this area, how can they get the training, get the skills, and not lose whatever that is they're bringing from their art perspective? So I think I'm going to close on that. Here's some shows that are coming up or have been in the last few years. So there's a lot of growing activity. But hopefully this just gives us a little bit of background on something that might be useful for later. Oh, one other thing in terms of access. I subscribe to every journal I can. I mean, these are like trade journals. These are given away free for people in the trade. So this is my biomedical products. And it was the same 20 years ago with computer stuff. You didn't have things like Wired or Byte magazine. You had Byte. But you had to go read the trade journals of the people in that industry. So this is very nice pictures. Those of you, the future is now. Where's some other ones? Fierce little UVs for a bio world. A ray of hope in quantitative analysis. I mean, it's just kind of an interesting world between a very specialized academic discipline and a more public thing. Thank you.
Thank you, Stephen. Did I steal your chair? Our next speaker is Cheryl Kerfeld, who's a research molecular biologist here at UCLA in the Lab of Structural Biology and Molecular Medicine. Cheryl holds an MA in English literature and a PhD in molecular biology. In addition to doing her research in proteomics, she has taught an interdisciplinary course entitled The Genome, Blueprint, Controversy, and Destiny. Just like Ruth, her students were a mix of humanists and science majors. The course explored a broad range of topics related to the Human Genome Project, with special emphasis on laboratory hands-on work. And I should point out that Ruth West, also teaching art and genetics, was very much teaching the fundamentals of genetics and introducing students to how they would hands-on work with this. So I think Stephen brings up an important point, that there's an issue with artists getting a glimpse and making metaphors and looking at it just from a surface point, because it actually creates more of a gap in the dialogue between art and science. So both Ruth and Cheryl play a really important role here on this campus by educating a broad group of interdisciplinary students to fundamental issues in the humanities and the arts that deal with our kind of larger context, while really teaching students some of the fundamentals of genetics. So please give a warm welcome to Cheryl Kerfeld.
Thanks. I can speak for at least one biochemist who would like to see a cactus grow hair. I haven't been so heavily socialized. So what I'd like to do, I guess, this was rather difficult. I mean, for me to sort of put together my interests as a protein biochemist and a crystallographer. So in other words, I'm in the business of determining what proteins look like, which is essential to understand how they function, which is something that's pretty wise to have in your pocket if you're going to start manipulating that protein and trying to have it do different things. So I'd like to talk about that a little bit and then tell you a bit about how that informs my other role here at UCLA, which is in designing undergraduate curriculum. And so with that beginning, I'd like to build on some of the things that Gregory talked about, and perhaps I think through the things that I want to sort of point you towards, sort of explain why things are moving so slowly. As he pointed out, not a lot's happened since that genome's been sequenced. And there's a couple of reasons for that I think will become apparent here. And then I guess what I'd like to do is to try to flesh out the picture a little bit more with the bodies and the structures of proteins to kind of give you a reason for why this is taking so long, and also give you some things, not so much to try to tell you what to think about these issues, but some of the things that we ought to consider, some of the things we ought to be thinking about. And this then too, of course, fits into my role in educating both those people who are going to be the bioengineers of next year, and also the year physicians and so on. So with that said, let's just sort of look at the really big picture here. And I'm going to show you or treat you to some pictures out of the textbooks that I teach out of. So we're accustomed to getting wonderful things like this. And the area that that my work in is determining the structures of individual proteins. Okay, so what we find out is that DNA is a really sort of monotonous, uninteresting, information-bearing molecule in terms of its shape. And I'm sure that's why that the audio, the music that you just heard that was macromolecular based, was based on proteins, which are very interesting, very sexy, have all sorts of different shapes and so on. Whereas DNA is very uniform and there's only 4 bases involved. And basically all of life, which is one of those things that we teach our students, well, you know, life is highly organized, is the interactions of proteins with one another, the interactions of proteins with one another to produce structures, as well as produce the other molecules that make cellular life possible. They glue cells together to form tissues and bring tissues together to form organs and so on and so forth. So really the fundamental thing which DNA encodes is the protein. And so there's this whole area of molecular biology, protein crystallography, protein structure determination, where we spend a lot of time trying to determine the structures of proteins, and it's very labor intensive. And that, I think, is one of the reasons for the big bottleneck in moving from the genome in terms of the DNA sequence into actually manipulating it. And one of the reasons why we ought to be hesitating a little bit before we jump into things, because there's lots we don't know. So just to sort of remind you of this, I think probably many of you have this in your knowledge base already, and if you don't, we probably all ought to with the coming policy issues and so on that we're going to be addressing in the next few years. But the gene or the DNA will give you a linear sequence of amino acids, and that's it. However, in order to get to functioning proteins, this linear sequence will fold up first in certain orderly local regions, and then this will fold up upon itself, and then in some cases you'll have multiple copies of folded proteins coming together. Again, if we understand and we know all the sequences of the genes in the genome, that's great, and we don't even have that yet. But then becomes the next large question is, what do these things look like? Okay, so again, if you're thinking about all this manipulation, all these things that we're talking about doing, we need to know the shape of the proteins and their interactions and so on. So again, we're a long way from some of these scenarios. I agree that it's not a question of if, it's a question of when, but for different applications, I think, we're at different distances from doing these sorts of things. And just to give you sort of a classic example of the sort of work that I do and why this is really important is that this is the way that we understand how things work. And in this particular case, this is the protein molecule that's on the surface of a flu virus that allows it to latch onto your cells and give you an infection. And so again, we find out that the shape, the structure of these things is intensely important for its function. And in fact, the two cannot be disentangled. You don't say structure follows function or function follows structure in this case. It's before the evolutionary split of those two things, It's the intertwining or the integration of structure and function. And now I want to just tell you a little bit about this particular work that I do. And I use actually proteins that come out of this organism, or I'm studying proteins that come out of this particular organism. And you might say, well, why? I'm here to talk about the human genome. Why are you telling me about this really cute Spirulina cyanobacterium? Well, the idea is how do we know what genes do? How are people who are busy annotating the human genome, how do they know what any segment of DNA does? Well, a lot of it is based on work with model organisms, and we've already heard a little bit about transgenic mice, You don't go out and start manipulating a piece of DNA that you're wondering about in a human being for the obvious ethical implications. But if you can fool around with mice and make them obese, as we saw a little bit ago, or if you can fool around, better yet, the proteins that you're interested in are found in something as lowly as this organism, where you're not going to get into any ethical conundrums when you think about manipulating it, all the better. So this is the particular model organism that I work with, because I study proteins that transfer electrons in cells. And if electrons weren't transferred in your cells, you wouldn't be here either. And I should also mention that this is spirulina, so this is something, if you go down to tropics over there, you can have a nice scoop of this on your smoothie. And so when I need more spirulina for my research, I don't just go down to tropics. That would be way too expensive, for this. But I can go out to Calipatria. I can drive out by the Salton Sea where this huge farm of spirulina growing in these vast lakes where they created, or they're making this just for this purpose, to supply health food stores and so on. And I can go out there and buy a couple of kilos of spirulina for $20 and then bring it back to the lab and grind it up. And when I grind it up, I might get a fragment of the inside, such as what you're seeing right here. And you're looking through an electron microscope at this point. And we're going to focus in now in our really sort of spirit of reductionism that's so central to the sorts of inquiry that we scientists are trained in, and this very tunnel vision thinking that we do. I'm going to zoom in on this particular area, and I'm going to show you the picture again that I would treat my students to out of the textbook. And what you see here is a protein, all these different colored blobs are proteins. And they don't really look like this. They're not really orange and yellow with words on them or anything like that, but we just don't know what their structures are. So we use this sort of gross oversimplification. Which has its benefits, but of course one thing that's important is to recognize the limitations of your metaphors and of your pictures and so on. And the particular protein actually that I'm going to zoom in on is this little one right here. And I can spend years, or I can actually tell you that I spent a good deal of my graduate career just trying to look at that protein using the techniques that we do. So you take this organism that you buy at the Salton Sea, you bring it back to the lab, and you pop some in the blender and grind it up, and then using various different physical separation techniques that you'd find chronicled for you in that trade magazine that you had there, you would eventually come out with different purified mixtures of proteins, or purified, I shouldn't say mixtures, purified proteins. So we've got just one particular protein in here. And then another huge bottleneck for us as structural biologists is coaxing that to form a three-dimensional crystal. Now again, let's not lose our focus here. The whole idea is, or why I'm bringing this all to your attention, is we're a long way from the gene in order to really understand how something functions. We have an awful lot of data right now, a lot of gene sequences, but we don't have very much knowledge about them, in depth. So I want to put that all in your heads a bit for thinking about what really is the prudent thing to be doing here, and what sort of policy issues and things should we be thinking about? So again, this whole area is called proteomics, okay, and understanding the structures and the functions of the proteins that are in the genome. Again, this is essential to doing any sort of careful manipulation. Again, I point out, because I'm using your tax dollars to fund my research, it's important that we work in model organisms, okay, that we use other things in order to try to obtain proteins that we can use for research like this. You're looking through the microscope right now. This is about 1 millimeter across this way, and this is a crystal of protein. All right, and so then eventually, after a couple of years, you determine the structure of the protein. Extremely labor-intensive, computational-intensive, and so on. And this is the point I want to make, because this happened to me. I guess I solved this about a year ago. And it makes a couple of points about the process of science that I'd like to share with you, because I think we've touched on some of them already. Certainly the economic factor in motivating research. But also there's a very human factor that I think artists can identify with. I think it's been said earlier, the passion, and we tend to follow things, you know, in this way. Well, scientists do it very much the same way, because I would argue that art and science are just extensions of human development as human endeavors, creativity, you know. So it's all the same, because I'm going to tell you about something sort of irrational I did with respect to this structure. First of all, much to my surprise, instead of this being just one chain, and they're color-coded here for you, what I found was that I had an assembly of 24 molecules that made this sort of, well, if you look at it, it's probably the most ecumenical structure you'd ever want to see. Here you've got your Star of David formed, and here you've got a Greek cross. So anyways, this is 24 molecules, which creates a chamber, all right? And this, although I had no idea what does it do, all right? What does it mean? You'd never know that this particular protein would assemble into clusters of 24 molecules from the genome sequence. Okay, that's not going to be in there. Yet it's so essential to know that to understand what it does functionally. Okay? And here's the other thing that's sort of the kicker, and this is a problem, and this is something that I think scientists are grappling with right now, is we've been so reductionist for so long. And when you get something like this, it really underscores that point for you. I can't get it to do this when it's by itself in the test tube. If I've just got this one particular protein purified in a test tube, I can't get it to adopt this configuration. It turns out, and here's the lesson in this, and even though I did a ton of experiments to try to get it to do that, I never could. And it's sort of, and I bring this into your attention, actually let me back up to a point here, is here's the structure I just showed you, only rendered slightly differently. Sometimes we like to use these tubes to represent the helical elements. But actually, it turns out that back in 1995, the structure that got me out of graduate school looked like an assembly of 2 molecules. And at that point, it had never, ever, well, this was the first structure of this sort, and we all agreed, or everyone in the literature said, this thing works by itself. It works as a monomer, as a single copy. And my structural data, which are shown right here, certainly argued otherwise. I had a really hard time getting this published, even the first time, because we have this sort of entrenched notions about things in science. Just want to share that with you, because I think it's really important about even how we have this whole metaphor of DNA buildup, of base pairing, and so on. I mean, are we sure we're even really seeing, right? Metaphors give way to new metaphors. Our paradigms change. Okay, this is a very minor example in the grand scheme of things. This is just what happened to me, but it stands for, or it symbolizes this problem, or this thing that we need to be aware of, is that we're going to outgrow our metaphors, and someday we may even marvel at the day when we thought of DNA as a double helix of complementary base paired things, because it just speaks to the limitation of the knowledge at that time. So anyways, I did a lot of experiments then when I eventually got to this point, and wasted a lot of students' time, you might say, to try to prove that this thing really forms. And it turned out a little bit about rethinking of this. So we looked at lots of properties of it, and here, this is just, I wanted to show you because I think I like the picture. If we change and we put every atom in, and we color, and we do this typically, it's like, you know how like milk is, like skim milk is light blue, and you have different colors for different kinds of milk. Well, we use the same thing in biology. Red means negatively charged, blue means positively charged, and white means neutral. So what I had here was this chamber, and here I've peeled away at the top piece of it to let you have a look inside, that that's very greasy and oily in the inside and very slippery with a lot of charges on the outside. So this is a really interesting structure. Again, no clue. What does it do, though? Okay, and I couldn't even get it to form this outside of in the crystals. So what one does is you go back and you start rethinking your picture of where this thing is. You've been so reductionistic, and you go back out and you think about, well, how do I really think about where this protein is? Well, I think it's just floating around in this sort of watery-like balloon here. And it turns out, and this is something that's really become appreciated in my field just in the last year or two, something we call macromolecular crowding. That if you crack open a cell, it's actually really quite organized, and there's a lot of stuff in there, and that they affect one another. And so it turned out that the moral of my story is I still don't know what that cluster's done. I've used a lot of people's hours and efforts and sweat trying to figure it out, but I can now get it to form that in a test tube if I add impurities to it, add a lot of other molecules to make it seem like it's crowded, okay? But what this required was this whole sort of backwards thinking about the biology. We tend to be very reductionist, take it away, isolate it by itself, and study it, when here, some of the biology is really predicated on the environment of it as well. So lesson, that's all I want to share. Now you say, wow, where does this go from here? Well, I've been asked by the provost, because of some of the different things that I have in my background, to do some work on undergraduate education, specifically science initiatives. And I think that I can tell you, and I want to share this with you, because, well, just like we're not moving so fast on manipulating the genome, the university is very similar in that these sorts of things get into education only very slowly as well. And that some of my work as a structural biologist certainly informs my work as an educator. So the sorts of things when I deal with undergraduate science majors, I think about the problems of oversimplification that I've just tried to sort of illustrate for you right there. The other thing that's very new, and of course the obvious things too, well, we need courses in bioinformatics now and so on. But I'm more interested in turning people onto the process of science. And also the process of the 21st century science, which is what we call discovery-driven, rather than saying I'm going to make a hypothesis and test it, and if it doesn't work, well, then I change my hypothesis. Case in point, I made a hypothesis, this thing formed, I couldn't get the data, what do? I try something else. I don't reject my hypothesis, I just pursue pig-headedly what I think is true, based on, you know what, really visual information in a lot of ways, right? Anyways, interesting ideas behind that. The idea, too, of having to synthesize and finding patterns in large amounts of information. Again, we have this rich amount of information from the Human Genome Project. And then here's the sort of thing where my humanities background comes in, is this idea of using writing as a way to foster some of these skills as well. And don't read all of this, but the idea here is just to get to you some of the points that we typically talk about in the process of science, is isolate this variable, control everything, and then reject the hypothesis if it doesn't work. Well, that's a lot of hooey. What we really do is we look at what question interests me, and then what do I think the answer is, okay? And then I pursue that, and hopefully something interesting will turn up along the way. And so in our course, when we talk about bias, it's not a dirty word. It's something that you know about yourself. It's sort of a Zen quality. It's like, I am this, it's okay, I'll just be it, all right? But I have to be aware of it. Okay, and so that's sort of some of the things that I think are rather interesting that I think the public sort of overlooks. And it's this idea, too, of organization and regulation of itself, and this ability to find out of this mass of data the meaning, okay, here as well. So we're looking at the genome sequence now. It's something like this. We don't have any shape to our language, really, only in a very rudimentary sense. But to actually get at meaning, we need protein structure. We need to actually understand a bit more of the rules by which these things work. And I also then, I just want to toss this in because I think it's an important point in this discussion, is that we're never looking at the phenomenon themselves. We're always looking at their consequences. We are extrapolating from a bands on gel, some of these little dark smudges in a jelly-like substance, to the entire you out there. Okay, there's a long way in between there, a big distance. Likewise, we can now do what are called DNA microarrays and gene expression profiles, where we can see what genes are turned on in certain conditions and so on. But once again, without the benefit of knowing what these things, these proteins that are made by these genes actually look like and who they interact with, we're still an awful long way from function. So I think my message in this is that we've got some very rudimentary detail here, but we're a long way from our bird there. Okay? And likewise, I just thought this fit here as well, too. It's so true about science in many ways. How can I know until I see what I say? Okay, so, and I try to tell my students that they have to learn how to write. And I also encourage them to draw as well, because I think you, of course, see lots of things the sort of close attention to detail, and also the fun of making mistakes. Okay, and what can happen from those mistakes. And I try to encourage that. I mean, evolution itself is a process of making mistakes that seem to fit better. And then I also point out to them that all this stuff that they're getting in the textbook, they're out there just copying the paradigms that I'm telling them and the picture I'm giving them of the universe. But of course, we're going to find out that this is rather incomplete, okay? And a bit of a simplification, and that I want them to learn this, but then we want to see them go on as well. Okay, and I just want to finish up a little bit here, because to remind you again, or this point that I keep making over and over, is right now we call the genome, okay, the blueprint of an organism. And that blueprint tells us where we came from. Well, I just ask you to consider the previous ideas of where we've come from, you know, before this particular model. Or here's one of my favorites, and both the girls and the guys can get in on this one. Preformation. Here you see a little baby in the sperm right here. And then this one, I just use this while being from Minnesota. I like to bring in lakes and Indians as much as possible, as unpolitically correct as that may be. But you see this idea of Eve carrying every organism, every subsequent daughter and son within her as well. So, and they get very tiny and tiny in this infinite regress on the butter box there. But then, the last thing I want to just very quickly tell you about is what I try to do, or what I'm working on for non-science majors curriculum in biology. Because again, I think if you want to appreciate a book, you don't have to be a novelist. If you want to appreciate a painting, you don't have to be a painter. But I think to really appreciate biotechnology and be able to work on it and vote on it, you have to have some fundamental understanding of what DNA is, so that you can be a good juror and a good shopper at Ralph's and so on. And so we're working toward design of non-science majors biology curriculum. And one of the projects that I have going with a group of people in philosophy and also other molecular biologists is a year-long cluster course called Biotechnology and Society, where we're going to look at some of these issues. And again, considering the shortfall of this metaphor of the genome as the blueprint of the organism, because everything is self-creating. When we start, we go from an egg to an embryo to an ego. Okay, here as you can see in my non-science majors course, this is actually an honors collegium. This student here is from math. This is a student from econ. This is a fraternity man and who's also studying history. Here's a young woman from world's arts and culture. And this course, what we tried to do is a laboratory-based course in which here you can see them isolating DNA, and we actually sequenced DNA in the class. And what this does is it sort of demystifies the whole process. And also, what I'm told is it encourages students to, you know, gives them the tools for sort of a lifelong learning about this, and really understands how far away from a blueprint, a two-dimensional blueprint, to an organism, to a person, to a voting member of our constituency, but what a big space that is in between. So that's sort of my overview of what I do. Thank you.
Thank you, Cheryl. Our last speaker is Natalie Bookchin, who's a media artist and educator at Cal Arts. Currently, she's a Guggenheim Fellow. And from 1998 to 2000, she was an active member of a collective called Artmark that you may be familiar with. She is about to release her newest project called MetaPet on April Fool's Day, which is an online computer game about a transgenic human Tamagotchi, produced with her collective group called Action Tech. Natalie also teaches art students genetics and culture and issues dealing with genetics and culture. So please welcome Natalie.
Well, I have to admit, I don't really teach them genetics. I don't think I can do it quite as well as you, but I do talk about the impact on the culture and what artists are doing. I'll just get to a site. Oh, I need. Thank you. Is that okay? Okay. So I want to start with a couple of phrases that I collected. The miracle of science, dramatically improving people's lives, transcending the limits of science, unlocking the potential of genetic diversity, understanding genes, creating cures. Life is our life's work. Discovery happens here. Focused on curing disease. Discovery in the living organism, making sense out of life, one genome, two arrays, infinite discovery, fueling innovation, driving discovery, making a map of life, a blueprint of health. Now, these phrases sound like they could be coming from the great scientists of our day, but in fact, they're coming from, these are the slogans of the top biotech corporations, who we have to remember when we listen to what they say, are beholden first to their shareholders and not to patients. And I want to start by, I've got notes all over the place from these very provocative speakers that went before us. And I want to start by reframing a question that I think hasn't been reframed yet. And that is that I don't think that, I don't see the genetic revolution as an object that's fixed and natural and outside, that is a thing that we need to negotiate as either good or bad. But I see it as a cultural construction that is something that some artists who interrogate culture and society are trying to understand where it came from, how it was formed, and sort of what are the, like basically looking at it as a story that we tell ourselves about life and unpacking it and exposing it. So I don't see this binary of genetic engineering is good or genetic engineering is bad at all. What I do see is the question of, to go back to Gregory's presentation, he spoke a lot about we. You know, we are, you know, this, we are going to have this revolution, and our lives are going to be changed. And the question for me is, who is we, and whose lives are going to be changed? And if, you know, is we everybody in the world, or is we a very small group of elite that may get some kind of specialized cat modified so that they won't have allergies to it? So this is just a question. And I want to, I appreciated Cheryl's talk a lot, and I think that the question that we can no longer, as people who are not scientists, no longer convincingly tell ourselves stories about the neutrality of science, which is something that many of us grew up to accept. Now, it's particularly difficult to accept the neutrality, not just of the science, but of where the research is going and who's funding the research, when we've reached a time in history where the role of science and role of business entrepreneur has merged in a way that it never has before, and that you have more scientists on corporate boards than you ever had in any other time. So how the research is chosen is really something that needs to be looked at in terms of who's footing the bills and who's picking which applications get through. This is both the case in the so-called private sector and in the so-called public sector, which I think are much more sort of loosely, or they're not such fixed spheres as they used to be. public and private, when people working in universities are getting private funding and people working start their own companies who are also getting public and private money. So the last time that we heard this kind of optimism about revolution and about sort of the world changing was a couple of years ago with internet companies, where we were told that this new economy would change everything. And it's similar with biotech in that we're told that we are going to no longer have disease, that we are going to live beyond the age that we lived before, assuming that we all lived to the age that we were set up to, that our genetics determined that we were going to live to, and that we're going to get rid of hunger if we go to the GMO kinds of the rhetorics around GMO, and we're going to have customized babies on demand. Now, like the internet stocks, a lot of the rhetoric around these kinds of promises is coming out of companies and based on venture capital. And the difference between these kinds of, between internet stocks and the stocks that are sort of coming out of the biotech companies is that the false promises they don't just hurt the pocketbook. Now, as I wanted to just show you, speaking about the blueprint of life, there's some pretty amazing companies out there. And this is, instead of Renaissance, we have Genaissance. And here you have a very common image that shows up a lot, Michelangelo image. And this is personalized medicine from genomics. And now, I had a very brief conversation beforehand with our respondent, and we were talking about how any self-respecting scientist these days wouldn't ever say that one gene equals one, like there's one trait that goes with one gene. But this idea that there is this equivalent between one trait and one gene is something that was believed a number of years ago when a lot of these studies were emerging, and it's something that's known as genetic determinism. And as James Watson, one of the two scientists who discovered the structure of DNA, says, the genes are the ultimate description of life, and they tell us what it means to be human. I think that when it's really good for interdisciplinary research and crossover. I mean, it's something that's pretty useful. But when scientists start to become philosophers and tell us what it means to be human by looking at our genetic code, I think that's something that we need to worry about a little bit. So basically, the scientists now know, and thus the rest of the population who reads the science papers also have some sense of this, that the sequence of DNA can code for many things, and many things that are absolutely unpredictable right now. Okay, so we don't know what some of them code for. Genes turn on and off, and they function differently in different cellular environments and also differently in different external environments. The statistic that Gregory mentioned about environmental studies not affecting humans in any way, I think you said, is pretty. I'd like to see that statistic, but I have some other statistics that I'll bring up in a little while. So as I was saying, most scientists know that this is not the correlation, but there's still, as Cheryl was saying, this reductionist model that's being applied to the research, that the model hasn't been updated. And so the problem is that if you are, again quoting Cheryl, reductionist and have tunnel vision and are driven by passion towards your goal, then there might be a little bit of a problem in that direction may be missing out on some other key factors. And I wanted to give 3 examples. And the first one is human gene therapy. Now, human gene therapy, a couple of years ago, we were told was going to cure disease. And it sounds great. I mean, I would love it for that to happen. I'm not at all, I wouldn't say that I'm a critic of genetic engineering. I would say that I'm wondering if I'm questioning the premise of it. Again, going back to what I said before. So far, no one has proven that gene therapy will offer permanent relief without adverse side effects. And there are several hundreds of experiments going on right now. Now, there were human trials. Okay, so there were clinical trials where people were injected with foreign genes. And a couple of years ago, in 1999, Jesse Gellinger, a 18-year-old young man at the University of Pennsylvania, died from the side effects of one of these. And since then, the trials have no longer been allowed on humans. But what happened was that after Gelsinger died, there were investigations by the FDA, Congress, and NIH, and they covered widespread misbehavior. And that researchers had failed to disclose 6% of 700 incidents in which there were adverse side effects. Now, why is that? I would never say, some of my best friends and some of my family are scientists. I would never want to demonize scientists. But the problem is that in some way, and this is the problem that sort of was stated in sort of New York Times, New England Journal of Medicine, and other sort of major non-extreme leftist journals, was that the scientists are under pressure to push these experiments forward because there are venture capitalists and there are stockholders who have vested interests. And so when humans become guinea pigs in these kinds of research in which we're still applying this older model of the one gene, one trait, so therefore the gene is predictable when it enters into a new environment, that becomes, again, not just a problem for the pocketbook. Okay, second mishap. This happened last March, according to New York Times and a couple of other journals. Parkinson's disease patients. Now, Parkinson's, there's a lot of studies about the genetic basis of Parkinson's disease, and there's a lot of attempts to try to alleviate this horrible illness. So what happened is a couple of, or several patients with Parkinson's disease agreed to volunteer for clinical trials, and they had holes drilled in their heads, and they had them injected with fetal tissue cells. Now, there were unpredictable side effects. Those were, the patients ended up chewing constantly, their fingers would go up and down, their wrists would swell, their arms would flap uncontrollably, and these were irreversible. So it's a little bit of a problem when these things get pushed forward, again, without knowing without knowing enough, which is, I think, something that was really nicely stated by Cheryl. So both of these tests really damaged scientific credibility. And as I said before, human tests were eliminated in in gene research, in gene therapy. So many scientists now believe that these therapies will be useful, but only for a very small, like a handful of very specialized diseases. And even Craig Venter, who was the CEO of the private corporation that was participating in this race to map the Human Genome Project, has stated such. He's stated that this is really not going to happen, and he's also left Celera. So second example is cloning. Okay, so we heard before that cloning was, something that we all have to grapple with, these ethical issues about cloning. But Stephen mentioned a project by Natalie Jeremijenko, which can kind of bolster what I'm saying here, and that is this project, the One Tree. Well, the One Tree Project. And what she did is she planted a series, I'm just going to reiterate what you said, she planted a series of cloned seeds, and she had them raised in an environment in which she kept in a lab environment. So they were all treated with the same conditions. And then she presented it during a show in New York called Paradise Now, which was a show about the cultural implications of genetic research. And as she puts it, her punchline in the show was that all of the trees were different. So cloning, again, it's like cells, using the model that we are code, that human or other living organisms are code, and we can be reprogrammed just through this so-called language or this code, is highly problematic. And this is an art project that sort of illustrates this by showing that when you clone, you're not making an exact duplicate. other examples that are scientific, it has now been determined, well, we were promised a couple of years ago, besides getting rid of disease and besides getting rid of aging, that we were going to have armies of 6 feet tall, blue-eyed geniuses. And of course, some of us are impatient and it hasn't happened yet and we're disappointed. But the other thing that's happening is that most clones, it's determined, exhibit developmental failure before or soon after birth. And apparently normal clones have kidney or brain malfunctions for the most part. Cloned mice, there was an article last week in the paper, cloned mice become obese without overeating. Even Ian Woolmut, whose name became well known because he cloned Dolly, it was made headline news, I don't know, a month or two ago that Dolly has arthritis. And he said, and I quote, "It is questionable whether there are any clones that are entirely normal." And all of this calls into grave doubt an already questionable premise, which was that cloning is plausible, safe, or can even produce exact copies. And it's true that we did have that cloned cat in the newspaper a couple of weeks ago, but the cat looks nothing like the original. And where this kind of genetic research is heading is towards, as I said before, towards a kind of very specialized elite group who want, for sentimental reasons, some kind of genetic reproduction of their original, which is not going to be the same, but also they want, there's going to be cats that are specially made so that if you have allergies, you can still have a cat because the allergen will be coated out of it. But who knows what other side effects will come from that. That's something that only time will tell. And speaking of time will tell, I'll give one more example of where this research was pushed ahead and where I think that Donna Haraway's idea that humans and animals are the line between the two are merging more and more, in that now humans are the guinea pigs for these kinds of tests, whereas in the past it was just mice. In 1994, the FDA gave the go-ahead to corporations to produce genetically modified organisms. That means inserting a gene inserting the gene of one species into another species. So it's different than breeding in that in breeding you have controlled environments and you're cross-breeding, but you're never transgenically breeding. You're never crossing into another species. So it's quite different. And the FDA gave the go-ahead for corporations to produce this and to release it into the environment, requiring no regulations, no testing except for 90 days with rats, and with there was no knowledge, and there still is no knowledge about what the long-term environmental or health effects will be. And as Cheryl was saying, again, we don't know about there's so many complicated bits of information, and we have information, but we don't have knowledge. So there is also no labeling. There's labeling in most of Europe, in Russia, in like across of the continent, there is labeling for genetically modified organisms, but in the United States there is none. And there's no plan for liability in case of unforeseen consequences. So another problem that's not specific but is more general is that what this is doing is it emphasizes fixing individuals rather than fixing society. And it's an ideological position rather than a fixed given that's basically saying that we need to put fault on the individual body, saying there's something wrong with it, it's misprogrammed and it needs to be reprogrammed, rather than looking at the social and the political causes of illness and premature death. And it is, I don't have a handful of statistics with me, but if you do a quick search on the internet, you will discover something that you probably already know, and that is that most illness, most disease, and most premature death is not caused by faulty genes, okay? But and even if you just thought about the first world, let alone the third world, and let's not, thinking about life expectancy. And I'll give you just a couple of examples that I wrote out. And one is the case of asthma. Since 1980, there's been an increase in asthma, a 75% increase in asthma, which is impossible to attribute to a change in disease. In fact, studies have shown that the concentration of the disease is in poor neighborhoods, in polluted neighborhoods, near toxic waste sites, power plants, and near congested highways. So those of us unfortunate enough to be living in Los Angeles have cause for concern and might consider that the effects of the environment are the things that are going to tick off whatever becomes a mutation in our gene, rather than sort of locating the possible propensity for a disease somewhere down the line. Now, the problem with this knowledge is that it's not sexy, and it's not something that the NEA funds. Instead, the NEA, I'm like, this is like Freudian slip. It really was. The NEA does fund this stuff. No. The NIH funds research into the genetic causes of asthma. I think the NEA in the past might have funded research into environmental causes if artists were working on it, but that's another story. They don't do that anymore. Well, another example that I pulled out of an article in the New York Times on Wednesday was that now we're all, as women, Ashkenaz Jews, we're very concerned about breast cancer, right? And the deaths from breast cancer have been increasing radically. There are not more Ashkenaz Jews than there were in the past, but they've been increasing radically. Now, the statistics that the New York Times gave were that 5 to 6% of women are dying from genetically caused breast cancer. That means 94 to 95% of women are dying from breast cancer that has nothing to do with genetics. So that's, I mean, if you turn the numbers around, 5 to 6% is a big number, but so is 94 to 95. Now, interestingly enough, Black women in the United States, despite slightly lower risk genetically than white women, are 25% more likely to die from the disease than white women. And that can't be explained by anything except for environmental causes. But the research is not going into that. The research is going into the 5% of the, is looking for, as the article, the headline of the article stated, the breast cancer gene. Now, the problem also is that if you have the breast cancer gene, 50% of the time you won't, if you're at high risk, 50% of the time you won't contract it. So the question that I wish the researchers were directing their attention to is what is in the environment that triggers this response that leads to the sort of the mutation in the gene that causes breast cancer. But that's not, it's so complicated, and that's not where, again, where the money lies. And so that's not where the research is going to go until people decide to vote, like look into it a little and vote about it. Okay, let's see. Okay, I want to just end by showing my latest project. And what I have is I have a game that I've been making, and it's a transgenic human virtual pet strategy game. It's going to be released, as Victoria said, on April 1st. And in the game, what I'm trying to do is I'm hoping to make the player feel, not just be told about, but to actually emotionally feel these issues that are affecting their lives. And the game emphasizes that biotechnology is about business first and science second. So the MetaPet is this creature that in the year 2006, not long from now, scientists took an obedience gene. Okay, they took an obedience gene from a dog and they inserted it in a human to produce a more cooperative, more loyal worker. Now, as is often the case when you insert a gene into another species, there is a side effect. In this case, the side effect was seemingly, supposedly aesthetic, and that is the growth of a dog-like tail on the MetaPet. But as is often the case, again, it was hailed as a worldwide success. And let me go to it. I don't want to tell you too much about it because it's a game with lots of surprises, and it's also not up yet. But let me just show. But let me finish. Let me show you what I have, and then you can ask. So the way that you play the game is you are a manager, and you work for one of three biotech companies. One is called Purgen. which is a genetics diagnostic device manufacturer. The second one is called Gene First, and it's a biopharmaceutical company. And the third company you can choose to work for is called Star DNA, and it's a gene therapy company. Now, all of the companies that you may work for are linked to real companies on the internet. And as the stocks fluctuate, right now they've been really low, and there's a question about whether they'll ever go up. But in any case, what happens is the metaped or your worker may get depressed if the stocks go down because their 401ks may go down. So you're put in this position where you're kind of rooting for the company, perhaps. And as a player, you need to overcome the obstacles that are imposed by the limitations of the pet's mind and body using state-of-the-art technologies like genetic screening tests, biochemical pharmaceuticals, and body surgeries. So what it's doing is it's basically looking at the use of these technologies in a Western tradition, which is sort of the following industrialization and Fordism and Taylorism, where the body is instrumentalized to become more efficient. So when the outside of the body is sort of, we've given up on the of making the outside of the body more efficient, now the gaze is towards the inside of the body and how to make that more efficient. And if you look at some of the repercussions or the research that's been brought to fruition, what you find, especially in agriculture, where there has been quite a bit of research that's turned into, that's been released from the lab and gone out into the society, that the kind of work that's being done is work that helps to make agriculture more efficient. Okay, it's not work that helps to feed the world despite the rhetoric, but it's actually helping to sort of produce, to create monocultures where you have one or two major corporations sort of in control of what used to be in the control of many small groups. So in any case, back to the game, what else can I tell you about it? The metapet, despite the insertion of the gene, is not completely finished as a product that is sort of obedient. And so you as a manager need to use these various, you can see these buttons on the top that, right now, this is just a picture of what the interface looks like. All of the activity takes place inside this area, and here there's a computer screen that gives you information about how your pet is doing, and up here you've got secondary buttons when you roll over things like the eye icon and the pill icon and the body icon. For example, if you roll over the eye icon, you can test your pet to see if it has any if it has any bad genes, and that will also affect not necessarily the illness probability, but certainly whether or not your pet will feel happy or not if it's detected, if it is test positive for a particular gene. You can also read its email, which is a similar kind of surveillance technology that's made available through communications technology. You can use pills to either stimulate or tranquilize it. And the goal of the game is to make as much money as possible. And you're competing with other managers, so you can see how other managers are doing in your company and in other companies. But I'll leave it with that, because I think that would be interesting for us to have a general discussion. And I just want to say that check out the site metapet.net on April 1st and play the game and help me debug it.
Thank you, Natalie. Before we break, I'd just like to mention, also for those of you who came a little late, the speakers were chosen very deliberately by Catherine and myself to cover a very broad spectrum of issues and approaches to the whole genome project that we're participating in on some level. Gregory Stock put forward broad issues that engage public debate and policy, and this is the topic of his upcoming book, Redesigning Humans. Stephen Wilson introduced us to the many creative ways artists respond to these issues, from the pure aesthetic approach to harsh criticism that Natalie demonstrated so well. And Cheryl Kerfeld talked about her own creative process from a scientific point of view and raised some issues in relation to humanities and sciences and to the educational slow response to creating interdisciplinary courses that educate artists and scientists and really bridge that gap. And finally, Natalie brought to the table issues of corporate approaches to the Genome Project and the kind of snake oil that's being sold to raise the stock and all of this and what all that means. We're going to take a break now, and please take a look at the work that's being shown in this room. It's mostly done by science students, some by students from our department under the direction of Ruth West, who will be a respondent when we come back. She will basically start our discussion by asking questions that were raised by our four speakers. So let's all take a break, have a snack, and come back in about 15 minutes. Thank you.